Welcome back to the deep end. Today, we are shifting our gaze inward. We often think of our bodies as these solid, singular entities, a self.
Right.
But biologically speaking, that is, well, it's a comforting illusion, really.
It is. In reality, you're an ecosystem. You're a walking, talking planet.
I like that. You're a fortress.
Ah.
And it's a fortress that is under constant, relentless siege.
Oh, absolutely. Surrounded by billions, trillions of entities that see you as a resource.
They want to breach the walls, plunder the resources, and set up shop.
It's less of a singular entity, more of a disputed territory.
Exactly. And that's our topic today. Infectious disease. But I want to be really clear right off the bat. We're not here to talk about, catching a cold or taking some vitamin C.
No, this isn't about home remedies.
We are looking at the clinico-pathological foundations of infection. We're digging into the evolutionary arms race between the host, that's us, and the microbe.
And that framing is so critical. If you just view infection as, bad luck, you miss the entire science of it.
You miss the story.
You miss the story. This is a sophisticated biological negotiation. I mean, sometimes it's a stealth mission, sometimes it's a full frontal assault.
And sometimes it's just a communication breakdown.
A diplomatic failure, yeah, between our own immune system and the organisms that are supposed to be living on us peacefully.
So to navigate this battlefield, we're synthesizing two absolute heavyweights of medical literature.
The foundational texts.
We have Robbins and Codron Pathologic Basis of Disease, which provides that grand architectural map, the systems, the breadth, the clinical side of things.
On the big picture.
The big picture. And we are pairing that with Underwood's pathology,
which I think of as the mechanics manual. It's so good at the grounded, mechanistic explanation of how and why these things actually happen at a molecular level.
I love that combination. Robbins give you the what and the where, the whole landscape of the disease. And then Underwood comes in and just drills down into the machinery, the how.
Together, they really do give you a complete picture of pathology. It's not a static list of diseases to memorize. It's a dynamic process.
Exactly. Now, before we open the gates, we have to do our necessary disclaimer.
Always.
What we're doing today is an interpretive synthesis. We are building a conceptual scaffolding for you.
A framework.
This is absolutely not a substitute for the textbooks themselves. If you're studying this for real, if you want medical mastery, you cannot bypass the act of reading.
You can't. You have to get in there, look at the slides, read the actual chapters, and, struggle with the material a bit.
We're here to give you the mental model, the hook to hang that knowledge on.
Precisely. Think of this as the orientation before you set off on the expedition.
Okay. Let's start by defining the antagonists, the invading army. In the popular imagination, germs are just, germs.
Right. It's all one big, scary category.
But in pathology, the distinction between these agents is completely fundamental to understanding the kind of damage they cause.
It is. We categorize them based on their structure, their complexity, and most importantly, how they interact with us, the host. And the spectrum it's just wild. It ranges from these submicroscopic molecules all the way up to creatures you can literally see with the naked eye.
Let's start with the category that, arguably shouldn't even exist. The one that seems to break the fundamental rules of biology.
Prions.
Prions.
Yeah, prions are they're so bizarre because they challenge the central dogma of life.
Right. We're all taught DNA makes RNA, RNA makes protein.
That's the flow of information. That's life.
So where do prions fit in?
They don't. Prions have no DNA. They have no RNA. They are just proteins.
So hang on. If they have no genetic blueprint, no code, how do they replicate? How can you catch a protein?
That's the terrifying and brilliant part. They don't replicate in the biological sense. They recruit. Or maybe they corrupt.
Erupt. I like that.
A prion is essentially a normal protein, one that's usually found in the brain, that has, for some reason, folded itself into an abnormal pathogenic shape.
A misfolded protein.
A misfolded protein. And when this twisted protein bumps into a normal, correctly folded protein of the same type,
it doesn't infect it. It changes it.
It physically forces the normal one to twist into the same bad shape. It's a conformational change.
It's not reproduction, it's conversion. That's like a zombie bite.
It's exactly like a zombie bite. It is a chain reaction of misfolding. One bad protein corrupts two corrupt four, and all of a sudden your neurons are filling up with these insoluble, toxic, clumps of protein.
And this is the mechanism behind diseases like Creutzfeldt-Jakob and mad cow.
Exactly. Proteinaceous infective particles. That's the formal name. They're acting as a kind of slow-motion, self-amplifying poison.
Incredible. Okay, moving up the ladder of complexity, we have viruses.
Right, and Robbins calls viruses obligate intracellular parasites.
That sounds very technical.
It does, but the key word there is obligate. They have no choice.
Meaning what?
Meaning they absolutely cannot reproduce on their own. They are metabolically inert outside of a cell. They're completely dependent.
So they're basically hackers.
That is a great analogy. A virus is just a packet of codesome DNA or RNA wrapped up in a protein delivery system.
Shell.
A shell, yeah. It drifts up to one of our cells, injects its code into the hard drive, and completely tricks the cell's machinery into manufacturing more viruses instead of, doing its normal job.
So the cell becomes a factory for its own destruction.
For its own destruction, yes.
And then, okay, we have bacteria. This is where we get into what most people think of as a germ.
This is where we get into actual autonomous life. Bacteria are prokaryotes. They're complete single cells with all their own machinery, but they just lack a nucleus.
And they're incredibly versatile.
Unbelievably so. They can live outside our cells, they can live inside our cells, in the blood, in the gut, on the skin.
As we're going to get into later, they aren't always the enemy, right?
Far from it. We are vastly outnumbered by bacterial cells in our own bodies. Most of them are essential. The line between our helpful microbiome and a deadly pathogen is often just a matter of location.
Interesting. Then we have fungi things like yeasts and molds. And finally, parasites.
And parasites are just a huge, sprawling category.
It's a big bucket.
A very big bucket. It ranges from single-celled protozoa, like the plasmodium that causes malaria,
All the way up to...
helminths, parasitic worms, yeah. And even ectoparasites like lice and mites. We're talking about complex organisms with their own organ systems that are living inside of us.
So that's the cast of characters. Prions, viruses, bacteria, fungi, parasites. Our mission today is to trace the timeline of an infection. We want to understand the invasion.
So step one is the beachhead. How do they get in?
Right. And this is where we have to sort of shift our perspective on the human body. We tend to think of ourselves as so vulnerable.
But we're not. The concept of normal is actually incredibly robust defense.
We're covered in armor.
We really are. Infection is the exception, not the rule. It only happens when one of those barriers is breached.
Okay, let's look at the outer wall, the skin.
The skin is a masterpiece of defensive engineering. It's not just a passive wrapper. It's a hostile terrain for microbes.
Hostile in what way?
Well, first, physically. The outer layer is thick, it's keratinized, it's made of dead cells, it's just tough.
And chemically?
Chemically, it's got a low pH, it's acidic, and it's covered in these fatty acids that actively inhibit bacterial growth.
So it's basically an acidic desert. If I'm a bacterium landing on someone's forearm, I'm pretty much in a wasteland.
You're in a wasteland. You can't survive there for long, and you certainly can't just dig your way through it. That's why the vast majority of skin infections require a breach.
A cut, a burn.
A needle stick, an insect bite. The barrier has to be broken for the bacteria. They can't do it themselves.
But biology always has exceptions, and Robbins mentions the schistosoma larva. This is just the stuff of nightmares.
It really is. The schistosoma fluke is one of the very few things that can assault the wall directly.
How does it do that?
It lives in fresh water. If you wade into infected water, the larvae find your skin and release these incredibly potent enzymes.
What do the enzymes do?
They chemically dissolve the adhesive proteins, the sort of molecular glue that holds your skin cells together.
So it brings its own breaching charge.
It literally melts a tunnel through your intact skin to get to the blood vessels underneath. It's terrifying, but it's also very rare. Most pathogens are smarter. They prefer the path of least resistance.
They look for the openings we already have.
Exactly.
The inner tubes. I mean, we are essentially donuts, right? We have a hole running right through the middle of us, which is the gastrointestinal tract.
And that presents a massive strategic vulnerability for us. We have to let the outside world in, we have to eat, but at the same time, we have to keep the pathogens out.
Okay, so let's play this out. I swallow a piece of lettuce that's contaminated with, say, E. coli or salmonella. What happens next?
First, it hits the stomach. And the stomach it's a sterilization chamber. The gastric acid in there is incredibly potent. Most bacteria that go in just disintegrate almost instantly.
It's an acid bath.
It's an acid bath. Then, if by some miracle they survive that, they hit the small intestine. And that is a gauntlet.
A gauntlet of what?
Viscous mucus, Digestive enzymes that dissolve proteins, bile detergents that just break down fats and membranes, and these antimicrobial peptides called defensins that punch holes in bacteria.
Wow. It seems basically impossible for anything to survive that journey.
It is very, very difficult, and that's why for most gut pathogens, the infectious dose is really high. You need to swallow millions of salmonella bacteria for just enough to survive the gauntlet and cause disease.
But again, evolution finds a way.
Always. Some organisms, like Helicobacter pylori, have evolved to just neutralize the acid.
Right, H. pylori, the ulcer bacteria, how does it do that?
It produces a specific enzyme called urease. Urease splits urea, which is in our stomach, into ammonia, and ammonia is basic.
So it creates a little alkaline cloud around itself.
A personalized spacesuit, exactly. That neutralizes the stomach acid right around it, allowing the bacteria to live comfortably in the stomach lining, a place that should be lethal.
There's a really fascinating note in Underwood about cholera and stomach acid that just highlights how important that barrier is.
Oh yeah, this is a classic example. Vibrio cholerae is actually very sensitive to acid.
So it should die in the stomach.
It should. In a person with normal stomach acidity, you have to swallow a massive dose to get sick. But some studies have shown that if you neutralize that stomach acid, let's say with antacids, or in people who just don't produce enough acid,
Achlorhydria.
right, the bacteria becomes 10,000 times more infectious.
10,000 times.
10,000 times. The difference between a mild stomach ache and life-threatening cholera can literally be the pH of your stomach at that exact moment. It's an incredible statistic.
Now, there is a very specific mechanism of entry in the gut that I want to unpack because it honestly sounds like something out of a spy novel. The M cells.
Ah, yes, the Trojan horse strategy.
So what's the deal with these M cells?
Okay, so the gut lining is designed to be impermeable, right? A solid wall. But the immune system needs to know what's on the other side. It needs intelligence.
It needs to survey the area.
It needs to survey the area. So we have these specialized cells called M cells or microfold cells that are sort of sprinkled throughout the lining.
And what's their job?
Their job is to reach out, grab samples of particles from the gut lumen, and hand them over to the immune cells, the Peyer's patches, that are waiting on the other side. They are the intake valve for the immune system's surveillance network.
So they're supposed to be the security guards checking bags at the gate.
Precisely. But certain pathogens like Shigella, Salmonella, even poliovirus have evolved to exploit this system.
Wow.
They have proteins on their surface that bind specifically to these M cells. They trick them. They effectively go up to the security guard and say, hey, inspect me.
And the M cell obliges.
The M cell does its job, transports them across the lining, and then deposits them right into the waiting tissue, right at the feet of the immune system. They use the security checkpoint to bypass the wall completely.
It's a brilliant, if devastating evolutionary adaptation.
It really is.
Okay, let's move to the respiratory tract. We breathe in thousands of liters of air every single day, filled with dust, pollen, microbes. Why aren't we constantly sick with pneumonia?
The mucociliary escalator.
I love that term. It sounds so mechanical.
It is machinery. The entire respiratory tree, from your nose down into your lungs, is lined with cells that produce a layer of sticky mucus.
So it's like flypaper.
Exactly like flypaper. It traps all those inhaled particles. But then underneath the mucus, the cells have these tiny hair-like projections called cilia.
And what do they do?
These cilia beat in a coordinated rhythmic wave. They're all beating upwards, constantly pushing that entire mucus carpet up out of the lungs and into the back of your throat.
Or we just swallow it.
And let the stomach acid deal with the mess. It is a constant, brilliant, self-cleaning oven.
But as we're seeing, this is a pattern. Pathogens have a countermove.
Always a countermove. The influenza virus, for example, has proteins on its surface hemagglutinin that allow it to grab onto the epithelial cells and stick so it can resist being swept away by the escalator.
And some are even more aggressive.
Oh, yeah. Bordetella pertussis, the bacteria that causes whooping cough, is much more aggressive. It doesn't just stick. It releases a toxin that specifically paralyzes the cilia.
So it hits the emergency stop button on the escalator.
It breaks the machine. It destroys the clearance mechanism. And that's why the clinical symptom of whooping cough is that terrible, violent hacking cough. The patient is trying to mechanically force all that mucus out because the biological clearance system has been completely sabotaged.
And sometimes we sabotage it ourselves.
Smoking. Cigarette smoke is incredibly toxic to cilia.
So it does the same thing as the pertussis toxin.
Essentially, yes. Chronic smoking first paralyzes and then eventually just destroys the mucociliary escalator. And that is why smokers are so profoundly prone to respiratory infections. They have voluntarily dismantled their primary defense system. A bacteria like Pseudomonas looks at a smoker's lungs and just sees a wide open door.
Okay, so we've covered the breach. The microbe is in. Step 2 is establishing a foothold. As Underwood really emphasizes, getting in is not enough. You have to stay. You have to stick.
Adhesion. This is absolutely critical. Imagine you're a bacterium in the urethra. A periodic flood of urine comes crashing down trying to wash you out every few hours.
Or in the gut, with peristalsis pushing everything downstream.
Right. If you don't have a way to stick, you're just flushed away. The infection is over before it begins.
So they have, what, molecular grappling hooks?
That's basically it. Bacteria have surface proteins called adhesins, or sometimes entire structures called pili or fimbriae that act like Velcro. But here's where it gets mechanistically so interesting.
Okay.
These aren't just generic glue. They are highly, highly specific keys.
And this specificity explains the concept of tissue tropism.
Exactly. This is the answer to the question. Why does the hepatitis virus attack the liver and not the heart? Why does the flu virus attack the lungs and not the skin?
Because the adhesins on the pathogen only bind to specific receptors on specific host cells.
It's a lock and key system, a perfect molecular match. The influenza virus's hemagglutinin binds to sialic acid, which is incredibly abundant on cells in the respiratory tract.
And HIV binds to the CD4 molecule.
Which is found almost exclusively on helper T cells.
So HIV doesn't destroy the immune system because it, has some grand strategy to destroy the immune system.
No, it destroys the immune system because the CD4 molecule is the only door handle its key happens to fit. That very specific initial interaction dictates the entire course of the disease.
Okay, so once they're stuck, they often need to move. They need to spread out from that initial beachhead. This brings us to section 2, spread and evasion, the cat and mouse game.
The spread can be local or it can be systemic. For local spread, a lot of bacteria act like a demolition crew.
What do you mean?
Bacteria like Staphylococcus aureus or Clostridium perfringens just secrete enzymes, things like hyaluronidase and collagenase, out into the tissue.
Hyaluronidase, that breaks down hyaluronic acid.
Which is the cement, the ground substance of our connective tissue.
So they're dissolving the matrix.
They are chemically liquefying the matrix that holds all the cells together so they can just swim through the tissue planes. It's not passive at all. They are actively carving a road for themselves.
And if they want to go further, go systemic.
Then they hop on the highways, the lymphatic system, and the bloodstream. This is how a little skin infection becomes sepsis. A local problem becomes a body-wide disaster.
But there's a third route of spread that I find particularly unsettling.
Neural spread.
Yeah. This is the route taken by rabies and some of the herpes viruses.
It's terrifying. Let's consider the rabies virus. You get bitten on the leg by an infected animal, the virus gets into the muscle tissue, but it doesn't stay there.
It finds a nerve.
It finds a peripheral nerve ending. It enters the axon of that nerve and then hijacks the nerve's own internal transport system. The retrograde transport system the nerve uses to move supplies from the periphery back to the cell body.
So it gets a free ride.
It gets a protected free ride all the way up the spinal cord and into the brain.
It uses our own internal wiring as a protected tunnel.
And it's protected because the immune system has very, very poor visibility inside a nerve axon. It is a completely stealth run straight to the central command.
Speaking of stealth, let's talk about evasion. Because once the invader is in and spreading, the host's defense forces, the immune system, finally wake up.
Yeah.
The cat starts hunting. So how does the mouse hide?
There are really three main strategies you see over and over. Hide your appearance, hide your body, or just fight back directly.
Okay, let's start with hiding the appearance, antigenic variation.
Or, more simply, changing coats.
Changing coats.
The immune system recognizes pathogens by their surface proteins, their antigens. Once the body makes an antibody against a specific shape, that pathogen is marked for death. It's toast.
So the pathogen changes its shape.
Some do. Pathogens like Borrelia, which causes Lyme disease, or trypanosomes, they simply have a repertoire of genes for different surface proteins. They express one for a while, and just as the immune system mounts a response,
You switch to a new one.
they switch to a new one. So the immune system spends a week making a perfect weapon against code A, and by the time it's ready to attack, the bacteria is already wearing code B.
And the immune system has to start all over again from scratch.
Exactly. This is what causes the characteristic relapsing fever you see in these diseases. The fever goes up as the immune system fights, then down as the bug is suppressed, then back up again as the new variant emerges.
But the undisputed master of this is influenza. This is why we need a new flu shot every single year.
Right, and influenza does this in two ways. There's antigenic drift.
Which is a slow one.
That's the slow, gradual accumulation of small mutations. The virus's replication machinery is a bit sloppy, so it makes mistakes. These mistakes change its coat just enough that last year's antibodies don't stick quite as well.
But then there's antigenic shift.
And that's the one that causes pandemics. That's the big one.
How does that happen?
That's when two completely different flu viruses, say a bird flu and a human flu, infect the same cell, usually in a pig.
The mixing vessel.
The mixing vessel. Inside that pig cell, they swap entire segments of their RNA. The result is a brand new Frankenstein virus with a completely novel coat that no human immune system on the planet has ever seen before.
Yeah.
That's how you get 1918. That's how you get global devastation.
Chilling. Okay, strategy 2. Fighting back. Resisting phagocytosis.
Our frontline soldiers are neutrophils and macrophages, and their job is to grab bacteria and eat them, phagocytose them.
Simple enough.
But bacteria like Streptococcus pneumonia or Haemophilus influenza have evolved to wrap themselves in a thick capsule, a slippery, slimy polysaccharide coating.
So it's like trying to grab a greased watermelon.
That's the perfect analogy. The phagocytes just can't get a grip. They slide right off. But my favorite mechanism, if you can have a favorite, is protein A.
Produced by Staph aureus.
Yes. And this one is just deviously clever. It messes with the antibody itself.
So how does that work?
Okay, to understand this, you have to visualize an antibody as a letter Y. The two tips of the Y, the hands, if you will, are what grab the bacteria. The stem of the Y, the handle, is what our immune cells look for to trigger an attack.
The handle is basically the eat me sign.
It is the eat me sign, correct. So what Staph aureus does is it releases protein A, which has a very specific property. It binds to the handle of the antibody, that FC region.
So it grabs the antibody and holds it backward.
It holds the antibody backwards. It coats its entire surface in the host's own antibodies, but they are all facing the wrong way. So an immune cell floats by, and instead of seeing an intruder covered in eat me signs, it just sees a wall of self-proteins.
It's an invisibility cloak made of the enemy's own flags.
It's absolutely brilliant. It's immunological judo.
Okay, third strategy. Don't fight, just hide. Hiding inside our own cells.
Intracellular survival. Some bacteria, like Mycobacterium tuberculosis, actually allow themselves to be eaten by macrophages.
They want to be swallowed. That seems counterintuitive.
It does. The macrophage swallows the TB bacterium into a little bubble called a phagosome. Now, normally the next step would be for the cell to fuse that bubble with a lysosome, which is basically a sac of acid and digestive enzymes, to kill the bug.
But TB has a trick.
TB blocks that fusion event. It produces proteins that prevent the lysosome from joining the party.
It locks the door to the acid room.
It locks the door, and so it just creates a safe little haven for itself inside the very cell that was sent to kill it. It can live and replicate comfortably inside the macrophage for years.
And then there's Listeria monocytogenes, which takes it a step further.
Listeria is an acrobat. It gets eaten, but then it produces an enzyme that dissolves the phagosome membrane entirely, and it escapes out into the cell's cytoplasm.
It breaks out of jail.
It breaks out, but then it does something even crazier. It builds a rocket.
A rocket.
It hijacks the host cell's own cytoskeleton, the actin filaments that give the cell its structure, and it polymerizes them into a long tail behind it. It creates propulsion.
So it's using the cell's own structural beams to build an engine.
Exactly. It rockets around inside the cell and then uses that force to literally ram through the cell membrane and into the next adjacent cell.
So it spreads from cell to cell without ever going outside.
Without ever exposing itself to the extracellular space where all the antibodies and other immune components are waiting. It's like a fugitive tunneling between houses to avoid the police patrolling the streets.
Unbelievable. Okay, let's move to section 3, the arsenal. How do bacteria actually cause the injury? We've talked a lot about how they survive, but how do they hurt us?
Robbins really divides this into 3 main buckets: direct cell death from replication, inducing a harmful host response, and of course, toxin production.
Let's focus on toxins, because this is where the biochemistry gets really potent and specific.
Yeah.
And we have to start with the classic distinction between endotoxin and exotoxin.
This is a classic board exam question for a reason. It's a vital clinical distinction.
So endotoxin.
Endotoxin is LPS lipopolysaccharide. And the key thing to remember is that it's a structural component. It is part of the outer membrane of all gram-negative bacteria. It's part of the wall.
So the bacteria doesn't actively shoot it at us?
No. It is mainly released when the bacteria dies and falls apart or maybe sheds little bits of its membrane. And our immune system has evolved to recognize LPS as a universal alarm signal. It screams, danger, gram-negative bacteria present.
And a little bit of alarm is a good thing.
A little bit is great. Low levels of LPS stimulate a healthy, appropriate immune response, but high levels, like in a massive blood infection in sepsis, cause the immune system to just panic.
The cytokine storm.
It's the cytokine storm. It leads to systemic vasodilation, your blood pressure plummets, you go into shock, and you get this horrible thing called disseminated intravascular coagulation, or DIC.
Where your blood starts clotting everywhere.
It starts clotting in all your small vessels, using up all your clotting factors, so then you start bleeding out from everywhere else. That is septic shock. And it's not caused by a specific poison. It's a system-wide catastrophic overreaction to the alarm bell.
Contrast that with exotoxins.
Exotoxins are totally different. These are proteins that are actively synthesized and secreted by the bacteria. They are precision weapons, sniper shots. They have very specific targets and very specific enzymatic mechanisms.
And many of them are so-called AB toxins.
The key and the poison. It's a great model. The B subunit is for binding, the key. It attaches to a specific receptor on the host cell. That binding event creates a doorway that lets the A subunit, the active toxic part, the poison, get inside.
Let's take an example, diphtheria toxin.
Diphtheria toxins? A subunit gets inside the cell, and its only job is to shut down protein synthesis. It finds and inactivates a factor required by the ribosome.
And if a cell can't make protein?
It dies. Instantly. It's a complete shutdown of the cell's basic functions.
But not all of them kill the cell, right? What about cholera toxin?
Right. Cholera toxin is a great example of a non-cytolytic toxin. It doesn't kill the gut lining cells. It enters them and enzymatically modifies a G protein that controls ion channels.
And what does that do?
It basically locks the on switch in the on position. It forces the cell to continuously pump chloride ions, and water follows out into the gut lumen.
So it's not tissue damage in the traditional sense. It's a physiological override.
A complete hijacking of normal cell function. The patient can die of profound dehydration, but if you were to look at their gut lining under a microscope, it would still be perfectly intact.
And then you have the neurotoxins, the famous pair, botulism and tetanus.
They're so fascinating because they're produced by related bacteria. Both are Clostridium species, but they cause the absolute opposite clinical effect.
So botulinum toxin.
Botulinum toxin gets into motor neurons, and it cleaves the specific proteins that are required for the release of acetylcholine at the neuromuscular junction.
And acetylcholine is the chemical signal that tells a muscle to contract.
Exactly. So if you block its release, you get flaccid paralysis, you go limp, the diaphragm stops working, and you can't breathe. This is why we use Botox for winkles. It's a targeted, localized flaccid paralysis of the facial muscles.
And tetanus. The opposite.
The exact opposite. Tetanus toxin travels up the nerves to the spinal cord, and there it blocks the release of inhibitory neurotransmitters, glycine and GABA.
And these are the brakes on your motor neurons.
They are the brakes. They're what tell your muscles to relax. So if you cut the brake lines,
the engine just revs out of control.
And you get spastic paralysis. Every muscle in the body contracts simultaneously and violently. It's powerful enough to break your own bones. Lockjaw is the classic sign. It's the same basic mechanism messing with neurotransmitter release, but targeting different neurons to produce opposite effects.
This segues perfectly into our next section, section 4.
Right.
Because sometimes the real injury isn't from the toxin or the bug itself. It's from us, the double-edged sword of the immune response.
This is the friendly fire concept in pathology, and both Robbins and Underwood emphasize this over and over. In many, many infectious diseases, the pathogen itself is relatively benign. It's the host's attempt to eliminate it that ends up destroying the organ.
Let's go back to tuberculosis.
Perfect example. The TB bacillus doesn't really have potent exotoxins. It doesn't dissolve tissue on its own. But because our macrophages can't kill it, remember, it blocks that lysosome fusion.
The immune system has to call for backup.
It calls for major backup. It initiates what's called a type IV hypersensitivity reaction, a cell-mediated response.
And this is what forms the granuloma.
Yes. The body essentially decides that if it can't kill the intruder, it will imprison it for life. It builds a wall of specialized macrophages, giant cells, and lymphocytes around the site of infection. This organized structure is the granuloma, or the tubercle.
Which sounds like a pretty good containment strategy.
It is, for preventing spread. But the center of that granuloma often undergoes a specific type of necrosis called infectious necrosis. It turns into this dry, crumbly, cheese-like mush of dead tissue.
And that destroys the lung.
That destroys the lung parenchyma. The cavities you see on a chest x-ray, the coughing up of blood, the fibrosis and scarring, all of that is the direct result of our own immune containment strategy, not the bacteria actively nibbling on the lung.
Hepatitis B is another classic example of this friendly fire.
It is. The hepatitis B virus gets inside liver cells, the hepatocytes, and it just lives there. It doesn't lysolize them or kill them directly. A person can be full of virus and actually have a pretty well-functioning liver.
So what causes the liver failure?
Our own immune system. The cytotoxic T cells recognize the infected liver cells as foreign, and they systematically kill them to try and stop the viral replication.
So paradoxically, severe liver failure is actually a sign of a strong immune response.
In many cases, yes. In fact, in what's called fulminant hepatitis, where a patient dies very rapidly of massive liver failure, it's because their immune response was just too effective, too aggressive.
And what about the people who become chronic carriers?
In those patients, their immune system is more tolerant. It sort of makes a truce. They have the virus, but they don't have the intense inflammation. They're still infectious to others, but they aren't sick in the same destructive way.
And then there's the really tragic case of molecular mimicry. This is a case of mistaken identity.
Rheumatic heart disease. It's the textbook example. It starts with a simple Group A strep throat infection.
Strep throat.
Your body, quite correctly, makes antibodies against a protein on the surface of the strep bacteria called the M protein. But by a cruel twist of evolutionary coincidence, that bacterial M protein looks structurally very similar to certain proteins found on your own heart valves.
Oh, no.
So the antibodies do a great job of clearing the throat infection.
And then they turn around and attack the heart.
The heart. You survive the sore throat, but 10, 20 years later, your mitral valve is scarred, stenotic, and destroyed. The whole negotiation failed because the immune system couldn't distinguish the enemy from the self.
Okay, let's make this all visual. As pathologists, when you look under the microscope, you're not just seeing bugs, you're seeing the aftermath of these battles. Section 5, morphologic patterns of infection. This is where Robbins really shines.
It is. Pathology is a visual discipline at its core. We can often tell who fought the battle just by looking at the debris field. There are 5 main patterns you see again and again.
Let's start with pattern 1, suppurative inflammation.
Which is the technical term. In the clinic, we might call it purulent, but colloquially, it's pus.
Pus, which is something everyone recognizes, but I suspect very few people actually know what it is. Is it just like bacteria fluid?
No, and that's the key misconception. Pus is actually a graveyard, and it's mostly our cells. When you look at pus under a microscope, you are looking at millions upon millions of dead and dying neutrophils.
Our suicide bomber white blood cells?
Mixed with tissue that they have liquefied with their enzymes and edema fluid. It's a neutrophil-rich battlefield.
So the very presence of pus is a specific marker of specific type of battle.
Exactly. It tells you almost unequivocally that you are dealing with what we call pyogenic or pus-producing bacteria. Typically, that means extracellular gram-positive cocci like Staphylococcus or gram-negative rods like E. coli. These bugs provoke a messy, wet, chaotic war. They attract neutrophils that just dump out enzymes and liquefy everything.
Which is why an abscess feels soft or fluctuant.
Right, because it's a bag of liquid death encapsulated by a fibrous wall.
Okay, pattern 2, mononuclear and granulomatous inflammation.
This looks completely different under the microscope. There's no pus, there's no liquid. Instead, you see a dense infiltration of lymphocytes, plasma cells, and macrophages. The tissue is often firm or even rubbery.
And this suggests a more chronic battle.
A chronic battle or an intracellular one. This is the histologic signature of things like syphilis, tuberculosis, or many viral infections. A syphilitic char is a hard chancre because it's packed with these mononuclear cells, not liquefied by neutrophils. And if you see a well-formed granuloma, that organized ball of macrophages, you should immediately be thinking TB or maybe fungi.
Pattern 3, cytopathic cytoproliferative. That is a mouthful.
It is, but it breaks down easily. Cytopathic just means cell damage, and cytoproliferative means cell growth. This is the viral signature.
Why viruses?
Because viruses live inside our cells, they tend to remodel the furniture. They cause specific changes to the cell's appearance.
Give me a visual.
Herpes simplex virus. It infects skin cells, causing them to swell up, detach from each other, and form blisters. And if you look closely at the infected cells, they often fuse together to form multinucleated giant cells, or they contain inclusion bodies, little visible factories of viral parts inside the nucleus or the cytoplasm.
Like the famous CMV owl's eye inclusion.
The classic owl's eye. It's a huge and large cell with a dark inclusion that looks just like an owl's eye staring back at you from the microscope.
And the proliferative part of that.
That's warts. HPV, the human papillomavirus, doesn't just damage the cell. It tricks the epithelial cells into dividing uncontrollably. So instead of killing the cell, it makes it grow a mountain of new tissue. That mountain of tissue is the wart.
Okay, pattern 4. Tissue necrosis.
This is maybe the scariest one. This is when the tissue just dies rapidly. And when you look under the microscope, it's like a ghost town. The outlines of the cells might still be there, but there are no nuclei. And critically, there are often very few inflammatory cells.
Why no inflammatory cells?
Because they haven't had time to arrive, or they've been killed as soon as they got there. This is the signature of incredibly potent toxins.
This is the toxin effect we talked about earlier.
Exactly. Clostridium perfringens, gas gangrene. The toxins produced by this bacterium dissolve the tissue structure so rapidly that it just turns into a chaotic, necrotic mush before the immune system can even react. You often see gas bubbles in the tissue from bacterial fermentation. It actually crackles when you touch the skin. This is a true surgical emergency.
And finally, pattern 5, chronic inflammation and scarring.
This is the stalemate, the long, drawn-out war that nobody wins. If the body can't clear the infection, but the infection can't kill the host, the end result is often fibrosis.
The body just paves over the problem with scar tissue.
It tries to. In the liver, the eggs of the schistosoma parasite can get lodged and cause this dense pipestem fibrosis. Chronic hepatitis B or C leads to cirrhosis, where the functioning liver cells are slowly replaced by useless collagen scar tissue. Ultimately, the patient dies not from the infection directly, but because their liver has been slowly turned into a rock.
We've covered the mechanisms and the patterns. Now let's just zoom in on a few specific pathogens to see how all of this comes together in practice. We can't cover the whole textbook, obviously, but let's hit a few of the giants.
We have to start with the king of pyogenic bacteria, Staphylococcus aureus.
Golden staph.
It is the ultimate pathogen. I mean, it's a Swiss Army knife of virulence. It has adhesins to stick to almost any surface skin, bone, artificial heart valves. It has a capsule to resist being eaten.
It has protein A to hide from antibodies.
It has coagulus, an enzyme that clots blood around it to form a protective shield. And on top of all that, it has a whole arsenal of toxins that punch holes in cell membranes and cause widespread damage.
Which is why staph infections can range from a simple pimple
to life-threatening osteomyelitis, bone infection, or endocarditis, the destruction of heart valves. It just respects no boundaries. If staph gets into the blood, it can seed an abscess literally anywhere in the body.
What about Pseudomonas aeruginosa?
That is the quintessential opportunist. It's famous for infecting burn victims and patients with cystic fibrosis. It absolutely loves wet environments.
And it has a very distinct look.
A distinct look and a distinct smell. In the lab, it has this weirdly sweet smell, like grapes or corn tortillas. But in a wound, it produces a blue-green pigment called pyocyanin. If you see a burn wound with blue-green pus, that is Pseudomonas until proven otherwise.
And it's particularly nasty because of its affinity for blood vessels.
Yes. It loves to invade the walls of blood vessels, causing a specific type of hemorrhagic necrosis called ecthyma gangrenosum. It's a very bad sign.
Let's shift gears completely. A parasite. Malaria.
Plasmodium falciparum, the deadliest form. It has a complex life cycle, but the part that makes us sick is when it infects our red blood cells.
And that's what causes the classic cyclic fevers.
Exactly. That's from the red blood cells all bursting in unison every 48 hours or so, releasing a new generation of parasites and a flood of inflammatory debris into the bloodstream.
But the real killer, especially with falciparum, is cerebral malaria.
And this is a purely mechanical tragedy. The parasite forces the infected red blood cell to produce these sticky knobs, these proteins, on its surface. And these knobs stick to the endothelial lining of the capillaries, especially in the brain.
Why does it do that?
To avoid being cleared by the spleen. The spleen is our blood filter. Its job is to pull out old or abnormal red blood cells. The parasite wants to stay out of the spleen, so it anchors the red blood cell it's hiding in to the vessel wall.
But when that happens in the brain,
It clogs up the microcirculation, it causes ischemia, a lack of oxygen. It is essentially a microscopic traffic jam in the brain caused by a single-celled organism trying to hide.
finally, before we wrap up, let's just briefly touch on the host factor, section 7, because the bug is only ever half of the equation.
It really is. The equation for disease is something like microbial virulence plus the dose, all divided by host resistance. And if that host resistance number drops to near zero,
Then virulence doesn't have to be high at all.
it doesn't. And this brings us to the world of opportunistic infections.
These are organisms that are basically harmless to a healthy person.
Completely harmless. A great example is Pneumocystis jirovecii. You and I, we have likely inhaled it. It's probably in our lungs right now. Our immune system keeps it completely suppressed without us ever knowing.
But in patient with HIV/AIDS,
As their CD4 T-cell count plummets, that immune suppression lifts. And the fungus, which has been dormant, just explodes in population. It fills the alveoli with this characteristic foamy, proteinaceous exudate, and the patient suffocates.
so it's not a new infection, it's an unleashed one.
It's an unleashed one. It's the same story with Candida causing thrush or Aspergillus causing invasive fungal disease. If you see a serious invasive fungal infection in a patient, your first question shouldn't be, where did they catch this? It should be, what is wrong with their immune system?
We have covered a massive amount of ground here. I mean, from the physical barriers of the skin to the Trojan horse M cells in the gut.
From the stealth of rabies traveling up a nerve to the brute force of gas gangrene dissolving muscle.
From the wet liquefaction of pus to the dry, hard fortification of granulomas.
It is a dense, dense topic.
So, synthesizing everything from Robbins and Underwood, what is the ultimate takeaway? What's the one thing to hold on to?
That pathology is the study of the interaction. Always. You cannot understand an infectious disease by just looking at the microbe in a petri dish, and you can't understand it by just looking at a healthy human. The disease is the battle.
It's the process.
It's the process. It's the timeline of invasion, negotiation, damage, and repair.
And it's a timeline that, on an evolutionary scale, never really ends.
And that really is the final provocative thought, the arms race. We develop a new antibiotic. Bacteria develop resistance plasmids and swap them like trading cards. We develop a vaccine. The virus drifts and shifts its coat. We are not the masters of this domain. We are just participants in a dynamic, ongoing, and ancient biological contest.
And sometimes we're not even the main character. We're just the vessel.
Just the vessel, the battlefield.
For those of you listening, I really strongly encourage you to now go open Robbins and Underwood. Look at the images in the infectious disease chapters. You need to see the owl's eye inclusion. You need to see the visual difference between the chaotic mess of an abscess and the orderly, walled-off granuloma.
The pictures make it real?
Use this audio as the map, but you really have to walk the territory yourself.
Absolutely.
Thanks for diving deep with us. Keep your barriers intact, wash your hands, and we will see you in the next episode.
Stay healthy.